Hi everybody and welcome back to our discussion of Chapter 7, the Respiratory System and its Regulation. So to get started, let's talk about what is the respiratory system. The main purpose of the respiratory system is to carry oxygen and to remove CO2 from all of our body tissues. This does this through four main processes. The first is pulmonary ventilation and the second is pulmonary diffusion. These both occur through external respiration. The respiratory system also helps us transport gases via the blood, and then is also responsible for capillary diffusion, which is known as internal respiration. So, breaking this down a little bit more specifically, pulmonary ventilation is the process of moving air into and out of the lungs. Our lungs have two main zones, the transport zone and the exchange zone. When air is inhaled, it's going to travel through the body. It'll come in through the nose or the mouth. Then it will go through the nasal concha, if it goes to the nose, then to the pharynx, the larynx, the trachea, also known as your windpipe, go through your bronchial tree, and then to the alveoli. A little bit more about how the lungs sit in the body. Our lungs are going to actually be suspended in our chest cavity via pleural sacs. You have your parietal pleura, which will line the thoracic wall, and then your visceral or pulmonary pleura, which attaches to the lungs. This creates a very smooth surface that will slide past itself and allow the lungs to move freely within the thoracic cavity. Because we have these pleura, the lungs will take up the size and the shape of the rib cage. Looking at the anatomy, you have the lung, the pleural sacs, diaphragm, and rib cage, and this kind of area that's been defined by the diaphragm up, and then the shape of the rib cage is going to determine airflow in and out of the lungs. And then this will help influence inspiration when we breathe in and expiration when we breathe out. Looking at inspiration, inspiration is an active process. This is going to involve multiple muscles to make it happen. The diaphragm is going to go from the up position and it will flatten down, creating space in the rib cage, and then your external intercostals will move the rib cage and sternum up and out. This will expand the thoracic cavity in 3 dimensions and expanding the volume of the thoracic cavity in the lungs. This movement is going to allow the lung volume to increase, which is going to cause a decrease in intrapulmonary pressure. Utilizing Boyle's law regarding pressure versus volume, this means at a constant temperature, pressure and volume will be inversely proportional. Therefore, if pressure goes down, volume must increase. Air will then passively rush into the area due to the pressure differences. Forced breathing is going to use additional muscles, like our scalenes, sternocleidomastoid, and pectorals, so the upper neck and upper chest region, and this will allow the ribs to even breathe in even further. So if you were to breathe in, and then I tell you to breathe in even more, you should be able to do that, and those are going to be those extra forced breathing muscles. Expiration, on the other hand, is a passive process. This means our inspiratory muscles are going to relax. This will cause the lung volume to decrease and the intrapulmonary pressure to increase, and then the air will be forced out of the lungs, kind of like if you were to squeeze an inflated balloon. We can also have expiration be an active process with forced breathing. This is going to cause the internal intercostals to pull the ribs down. It will also use your latissimus dorsi and quadratus lumborum. These are really big, beefy back muscles that allow for this big contraction to force air out. And then your abdominal muscles are going to contract to help force the diaphragm back up into place. So the next series is going to take you through the process of breathing and what's happening. So this is a resting position for your diaphragm and thoracic cage. Then go into an inhale. You notice the diaphragm comes down and flattens. This will cause the pressure change and air rushes in, rib cage goes out. And then when we exhale, diaphragm comes back up, pressure increases, air flows out. So, looking more at expiration, we also have the respiratory pump. We mentioned this back in chapter 6. This is because we're having these changes in intra-abdominal and intrathoracic pressure. This is going to promote venous return to the heart, specifically with the superior and inferior vena cava. So as that pressure increases, venous compression is going to occur, and then when that pressure decreases, we're going to allow more venous filling. This is causing the milking action very similar to the muscle pumps that we talked about in Chapter 6, because we're having this constant pressure change that will assist with that right atrial filling. The next thing we need to be concerned about are pulmonary volumes. In an anatomy course, we would really focus on what these are. For the most part, what we are concerned with exercise physiology is more the spirometry measurements. So lung volumes, capacities, and flow rates. We may also assess somebody's tidal volume, vital capacity, residual volume, and total lung capacity. This is typically used as a diagnostic tool for respiratory disease. If you are interested in a career path like pulmonary rehab, you will pay attention to these, or if you're working with an athlete with, say, exercise-induced asthma. But for the most part, we just have kind of baseline measurements in exercise physiology for these. So this is a nice summary of the different values that you would assess. So your total lung capacity is basically the sum of your vital capacity and residual volume. Your vital capacity is the greatest amount of air that can be expired after a maximal inspiration. Your tidal volume is the amount of air entering and leaving the lungs during a normal breath. The functional residual capacity is the volume of air remaining in the lungs after a normal expiration, and your residual volume is the amount of air remaining in the lungs after maximal expiration. So to measure these, we would take a spirometer. As you can see, this gentleman is using here in the picture. Typically, we'd ask you to hold your nose, or we'd clamp the nose for you, and we would measure all of these volumes via the spirometer. The next topic we need to discuss is pulmonary diffusion. So this is when we have gas exchange between the alveoli and the capillaries. So we inspire air going through the bronchial tree to the alveoli, and then we have the blood path, which is going to go from the right ventricle to the pulmonary trunk, to the pulmonary arteries, to the pulmonary capillaries. Each of our individual alveoli are going to be surrounded by a capillary bed, and this is where gas exchange will occur. There are two main functions with pulmonary diffusion. The first is to replenish the blood oxygen supply, and the second is to remove carbon dioxide from the blood. So at rest, our lungs are going to receive somewhere between 4 to 6 liters of blood per minute. So if you remember, our typical cardiac output is around 5 liters per minute, so this makes sense. Our right ventricle cardiac output is going to equal our left ventricle cardiac output. That's because our lung blood flow is going to receive the same amount as our systemic blood flow. However, our pulmonary blood flow is going to be considered a low pressure circulation. So our lung mean arterial pressure is around 15 millimeters of mercury, as opposed to aortic mean arterial pressure, which is around 95. Since there is a very small pressure gradient, so between 15 to 5 millimeters of mercury, resistance is going to be much lower, and because we're dealing with much thinner vessel walls here. All right, so diffusion is going to occur across the respiratory membrane. You may also hear this referred to as the alveolar capillary membrane. And this is basically just the connection of an alveolar wall to a capillary wall, and then the respective basement membranes that kind of make these two stick together. There is going to be a very large surface area where these gases have exchanged, but it's very, very thin. We're talking somewhere between 1 to 3 cells thick. This allows for the maximization of gas exchange. It's a very efficient and quick system. So, how do we tell how much oxygen and CO2 is getting through this diffusion process? Well, we're going to take a quick step into physics. This is all going to rely on the partial pressures of gases. So we know that air, what we breathe in every day, is around 79% nitrogen, around 21% oxygen, and 0.03% CO2. When we combine all of this together, this is going to create atmospheric pressure. Then if I take each of those individual percentages, that's going to give us a partial pressure. We know this works because of something called Dalton's law. That says our total air pressure is going to be equal to the pressure of nitrogen, the pressure of oxygen, and the pressure of CO2. So if I take that information, and knowing that 1 atmosphere of pressure is 760 millimeters of mercury, that means I can figure out that the pressure of nitrogen, which would be 760 times that 79-ish%, will give me about 600 millimeters of mercury, and we can go on through and figure out the pressure contributions of each of these gases. So this will matter because we take Dalton's law and we use it with Henry's law. So Henry's law tells us the gases that dissolve in liquids, and they are going to dissolve in proportion to their partial pressures. So this typically depends on a specific fluid medium and the temperature. However, we make the assumption that the solubility of blood is constant at a given temperature. This means our partial pressure gradient is going to be the most important factor for determining gas exchange. So our partial pressure gradients are going to drive gas diffusion. Without a gradient, gases are in equilibrium and there is no diffusion. So we know that oxygen has a higher pressure than CO2. It tends to push into the bloodstream. So oxygen exchange. We did the math earlier. We know atmospheric partial pressure of oxygen is around 159 millimeters of mercury, and we know that the alveolar pressure of oxygen is around 105. We also know that the pulmonary artery pressure of oxygen is around 40. This means I have a pressure of oxygen gradient across the respiratory membrane. And it's somewhere around 65 millimeters of mercury. And then the pulmonary vein is going to be around 100. So these pressure differences in the different areas of the anatomy are going to be wanting to force air in. Another law that will be important to oxygen exchange will be Fick's law. This is the rate of diffusion that is proportional to the surface area and the partial pressure of the gas gradient. So, for example, we know the pressure of oxygen is around 65 and the pressure of CO2 is around 6. So since we have a much larger PO2 gradient, it is going to move much more efficiently than CO2. Diffusion constants are also going to influence the diffusion rate, and each constant will be different for different gases. So the pressure constant for oxygen is going to be lower because of constant oxygen movement, and the constant for carbon dioxide is actually going to be lower than it is for oxygen. So even though it has a lower pressure gradient, it will diffuse easier. So our oxygen diffusion capacity, we know the oxygen volume diffused per minute per millimeter of mercury of that gradient. And we know the gradient can be calculated from the capillary mean PO2, which will give us around 11 millimeters of mercury. So our resting O2 diffusion capacity is going to be around 21 millimeters of oxygen per minute per millimeter of mercury, which will give us a total of 231 millimeters of oxygen per minute for 11 millimeters of mercury gradient. When we're talking about maximal exercise, however, our venous oxygen rate is going to decrease, so we will have a bigger pressure of oxygen gradient. This means my diffusion capacity can increase by as much as 3 times resting rate. So at rest, our oxygen diffusion capacity is limited due to incomplete lung perfusion. So when I go to breathe in at rest, I'm not using the full capacity of my lungs. I'm only using about the bottom third of our lungs, and only that bottom third is going to be fully perfused with blood. The top 2/3 of my lung surface area is going to have poor gas exchange. However, as I start to exercise, this O2 diffusion capacity is going to increase because we're getting more lung perfusion. So my systemic blood pressure will increase, and this is going to open the availability of the top 2/3 of my lungs for perfusion. This means gas exchange can now occur over the full lung surface area instead of just the bottom third. Carbon dioxide, on the other hand, we talked about these big pressure gradient differences with oxygen. Carbon dioxide does not have nearly as large of pressure gradient differences. The pulmonary artery pressure of carbon dioxide is around 46 millimeters of mercury, but our alveolar PCO2 is only around 40. This means diffusion will be only permitted by a 6 millimeter per mercury pressure of CO2 gradient. However, we know that CO2's diffusion gradient is 20 times greater than that of oxygen. So even though we have a very small gradient, diffusion can still occur. Another thing we need to be concerned with when we talk about the perfusion and exchange of oxygen and CO2 is something called an oxygen cascade. So an oxygen cascade is the dropping off of our partial pressures of oxygen at sea level from dry ambient air to the tissues and then to venous circulation, draining those tissues. So every time we interact with a certain level of tissue, we're going to start losing oxygen. So if you see in the example over here, we start with about 159 millimeters of mercury when we have dry ambient air. Then when we breathe this air in to just the trachea, we're going to lose about 10 millimeters of mercury of pressure. This is due to humidification. We have to add moisture to that dry air. Then, once we go from the trachea to the alveoli, we're going to drop even further. This is because the oxygen has now been diluted by the partial pressure of carbon dioxide. Then as it goes to the pulmonary capillaries and the arterial blood, we have a small drop-off, but nothing significant. And then as we send it out to the body to be utilized and pulled out of the bloodstream by tissues, we're going to have a big drop-off. So how do we actually transport oxygen in the blood? The carrying capacity, as we've discussed earlier in Chapter 6, is around 20 milliliters of oxygen per 100 milliliters of blood, which means we can get about 1 liter of oxygen into about 5 liters of blood. And we know 5 liters of blood is our standard cardiac output. So, of the oxygen that we are consuming and bringing into the body, about 98% of this is going to be bound to hemoglobin in our red blood cells. Once we add oxygen to hemoglobin, we refer to it as oxyhemoglobin. If it's just hemoglobin alone with no oxygen present, we call it deoxyhemoglobin. Then we have about 2% of oxygen that just passively dissolves into our blood plasma. So when we're talking about the transport of oxygen, it's mostly going to be carried around by hemoglobin, a little bit into the plasma. Looking at the hemoglobin saturation, this is going to depend on the pressure of oxygen and the affinity or the want to bind together between oxygen and hemoglobin. When you have a high pressure of oxygen, such as what you would see in the lungs, the loading portion of the oxyhemoglobin dissociation curve is going on here, and we will have very small changes in hemoglobin saturation per millimeter of mercury change in PO2. When we get into low pressures of oxygen, such as what we're seeing in the body tissues, this is the unloading portion of the oxyhemoglobin dissociation curve. Well, then we are going to see large changes in hemoglobin saturation per millimeter of mercury change in the pressure of oxygen. So this is your oxyhemoglobin saturation curve. So when you look at it from the top to the bottom, we see this upper portion that is the loading portion. No matter how much I change the pressure, there's going to be a pretty high amount of oxygen loaded onto our hemoglobins. However, as we start to branch into the unloading portion, saturation is going to change quickly even if I only have small changes in pressure. And this is because we are allowing oxygen to unload into our tissues. So what is going to impact hemoglobin saturation? First is blood pH. When our blood becomes more acidic, our oxyhemoglobin curve shifts to the right. This is referred to as the Bohr effect. This means more oxygen can be unloaded at acidic exercising muscles. So even though the muscle is becoming more acidic, oxygen wants to get off of the hemoglobin faster, which is going to help us go through better muscle metabolism. So more acidic shifts to the right, more oxygen is offloaded to work with working muscle. Blood temperature will also cause a rightward shift. The warmer the body is, the more the oxyhemoglobin curve shifts to the right. This also means that we will have more tissue unloading during exercise. So as you can see here, this is the changing pH. The red curve is a standard oxyhemoglobin curve. And then as we decrease the pH, you see the curve shift to the right. If we increase the pH, we see the curve shift to the left. And this is our example looking at the temperature of the blood, red being our standard. Increased body temperature moves to the right, decreased body temperature, it moves to the left. So our blood's oxygen carrying capacity is going to be dependent on blood hemoglobin content. We also know that our blood's oxygen carrying capacity is defined as our maximum amount of oxygen blood can carry. So knowing this is based on our blood's hemoglobin content, we know that our hemoglobin will be saturated around 98 to 99% at rest with a 0.75 second transit time. So this exchange of oxygen onto hemoglobin is very, very quick. However, as we go through physical activity, these saturations are going to start to lower, and that is because the transit time is even shorter, because the blood is moving through the vessels so much faster. So we know 1 gram of hemoglobin will bind to 1.34 milliliters of oxygen, and we know our blood capacity is somewhere between 16 to 24. This doesn't really matter until we start worrying about individuals who have anemia. When an individual has anemia, they have a decrease in hemoglobin content, which means they're going to have a decrease in oxygen carrying capacity. Now, you can have anemia for a couple different reasons. There is sickle cell anemia, which is having red blood cells that are misshapen, so they don't carry oxygen as well. You can also have anemia because you have low iron content. This is very common in females. You can also just have pernicious anemia, which means you're just generally anemic and don't carry oxygen well. So there's a lot of different reasons why you can have anemia. However, once you have anemia, you have a lowered hemoglobin content, you don't carry oxygen as well. So let's look at carbon dioxide transport, because this is the second half of the equation. We know carbon dioxide is released as a waste product from cells with metabolism. However, unlike oxygen, which pretty much is almost exclusively carried by hemoglobin with a little bit of plasma, carbon dioxide has 3 ways to be transported. The first is as a bicarbonate ion in our blood buffer system. The second is to be dissolved in plasma, and the third is actually to be bound to hemoglobin. However, we refer to this as a Carbaminohemoglobin. So looking at the bicarbonate ions, this is how we transport somewhere between 60 and 70% of the CO2 in our blood back to our lungs. So when CO2 interacts with the water found in our blood, it's going to form carbonic acid. This is going to occur in the red blood cells, and then it will be catalyzed by carbonic anhydrase. Now, carbonic acid, however, doesn't want to stay as an acid, so it will dissociate into bicarbonates. So CO2 plus water equals that carbonic acid, which will then dissociate into a carbonate ion and a hydrogen ion. This hydrogen ion will then bind to hemoglobin as a buffer and trigger the Bohr effect. These bicarbonate ions will then diffuse from our red blood cells into the plasma. We'll then have 7 to 10% of the CO2 that just dissolves directly into plasma compared to the 2% with oxygen. When the pressure of CO2 is low, like what it is in the lungs, the CO2 will come out of solution very quickly and diffuse out to the alveoli. And then finally, the Carbaminohemoglobin. This is about 20 to 33% of carbon dioxide transport. This does not compete, however, with oxygen hemoglobin binding. Oxygen likes to bind to the heme portion of hemoglobin, and carbon dioxide likes to bond to the globulin portion of hemoglobin. So they don't fight each other. The hemoglobin state, the pressure of CO2, and the affect or the want of CO2, the hemoglobin binding, is going to have an interaction. So deoxyhemoglobin binds CO2 more easily than oxyhemoglobin does. So hemoglobin that doesn't have oxygen already attached to it wants to pick up CO2. That means as the pressure of CO2 increases, it's easier for CO2 and hemoglobin to bind together. And when the pressure of CO2 decreases, it's easier for CO2 and hemoglobin to break apart. Another big concept that's going to be important is the gas exchange of the muscles. And the way we know this is occurring is we measure the arterial-venous oxygen difference. So this will be the difference between the arterial and the venous O2 concentrations. This is shortened to an AVO2 difference, and this is going to reflect tissue oxygen extraction. So as extraction increases, venous O2 will decrease, therefore we increase our AVO2 difference. Our arterial oxygen content typically is around 20 milliliters of oxygen per 100 milliliters of blood. And then our venous O2 content is actually going to be mixed with what oxygen wasn't offloaded. So at rest, this tends to be around 15 to 16 milliliters of oxygen per 100 milliliters. So we see there's only an AVO2 difference of 4 here. So 20 minus 16 equals 4. However, with heavy exercise, we can take our AVO2, the mixed venous O2 content, to 4 to 5 milliliters of oxygen. So 20 minus 5 means I have an AVO2 difference of 15. So this shows that I have an increase in extraction due to physical activity. So this is just a visual representation of what I just discussed, and kind of shows the math that I went through, or kind of talked through. Oxygen transport to the muscle. We know that oxygen will be transported in the muscle by a very similar protein called myoglobin. It'll be similar in structure to hemoglobin, but it has an even higher affinity for oxygen than hemoglobin does. So this means hemoglobin wants to offload it at the tissue, and then myoglobin will gobble that oxygen up, and it doesn't want to let go. So this oxygen to myoglobin dissociation curve is going to be shaped very differently than the hemoglobin curve. So at the specific pressure of oxygen is going to be somewhere between 0 to 20, and it's going to be very, very steep compared to our hemoglobin dissociation curve. So this means that our loading portion for PO2 is going to be around 20, and our releasing portion is going to be around 1 or 2 millimeters of mercury. So as we can see here, this is a comparison between hemoglobin and myoglobin. Hemoglobin kind of has this very gentle S shape to it. Myoglobin, though, is a very stark kind of boomerang shape. Myoglobin does not want to let go to oxygen until it absolutely has to. So what will influence oxygen delivery and uptake? So we know the oxygen content of the blood matters. This will be represented by the pressure of oxygen and the hemoglobin percent saturation. This will create the arterial PO2 gradient for tissue exchange. We also know blood flow matters. So when we decrease blood flow, we decrease the opportunity to deliver oxygen to the tissues. So exercise should increase blood flow to our working muscles. So we talked about this in chapter 6, how we can redirect blood flow. We also know local conditions matter, such as the pH and the temperature. So we can shift the oxygen hemoglobin dissociation curve to the left or to the right based on pH and temperature. So we know when there's a decrease in pH or an increase in temperature, it tends to promote oxygen unloading in that particular. So how do we actually remove carbon dioxide? So CO2 will exit the cells by simple diffusion, nothing fancy, but its exit is going to be driven by the PCO2 gradient. So at muscle tissues, the pressure of carbon dioxide is high, and in the blood, the pressure of CO2 is low. So we're quite literally forcing the CO2 out of the tissues from high to low concentration grade. So with all of that in mind, how do you regulate pulmonary ventilation? So the body needs to maintain a homeostatic balance between the blood's pressure of oxygen, pressure of CO2, and the blood's pH. This is going to require coordination between the respiratory and cardiovascular systems. Typically, this coordination will occur via involuntary regulation of our pulmonary ventilation. So we have a few central mechanisms of regulation within the body. The first being our respiratory centers, the inspiratory and expiratory centers of the brain. Typically, these are located in the brainstem, particularly the medulla oblongata and the pons. This will establish the rate and the depth of breathing via signals to the respiratory muscles. However, our cerebral cortex can override these signals if necessary. So I can make myself hold my breath. I can make myself hyperventilate. So I can kind of override those signals to a certain point. I also have central chemoreceptors. These will be stimulated by an increase in CO2 in the cerebral spinal fluid, and this will increase the rate and depth of breathing and remove excess CO2 from the body. We also have peripheral mechanisms of regulation, so out in the body, that it will tell our brain what to do. We have our peripheral chemoreceptors, so particularly in our aortic bodies and our carotid bodies. These are going to be very sensitive to blood PO2, PCO2, and hydrogen ion presence. We also have our mechanoreceptors, which are going to measure stretch, and the pleura, bronchioles, and alveoli. When we have excessive stretch, we're actually going to reduce the depth of our breathing. This is referred to as the Herring-Brauer reflex. So finally, this is kind of a summary slide that walks through the inspiration, expiration, control mechanisms. This would be a great place to pause your video and take some notes. With that said, that is the end of chapter 7. I look forward to seeing you guys again for chapter 8 and the end of the module.